the fundamentals of genomic DNA isolation. So what is genomic DNA? Genomic DNA are present in all organisms. That is present in bacteria, that is present in plant, animals, and it is also present in viruses. So these are the major kingdoms. So wherever there is a living organism is there, for all living organism there will be a genomic DNA will be there. For some organism the genomic part will be RNA. So generally we can say nucleic acid isolation. So in the nucleic acid isolation there are two types. One is DNA isolation and another is RNA isolation. So this DNA also, when you see a genomic DNA, bacteria will be having two types of DNA, that is chromosomal DNA then plasmid, that is extra chromosomal DNA. So bacteria will have chromosomal DNA and an extra chromosomal DNA that is called plasmid. So in plant you will say nuclear DNA, that is which is present within the nucleus. Then there will be a DNA which is present in mitochondria. So mitochondrial DNA. There will be also some amount of DNA which is present in chloroplast. So that is chloroplast genome. In animals, you can find nuclear and mitochondrial. In viruses, DNA, that is the genomic DNA which is used. Suppose if it is a virus, you may find a RNA as nucleic acid. So here we are seeing a DNA isolation protocol. So when you say a DNA isolation, it is actually total DNA isolation, because in bacteria if you see, you are isolating all the plasmids and chromosomal DNA together. Plant also you are isolating nuclear, mitochondrial and chloroplast DNA together, and in animal you are isolating nuclear and mitochondria together. So even though you generally call it as DNA isolation, you are actually isolating the total DNA which is present in all these compartments. So this is called a total DNA isolation. Suppose if you want to isolate DNA which is present in nucleus, mitochondria and chloroplast separately, so first you have to separate those organelles, then you have to do the isolation. Otherwise the DNA what you isolate will be combination of all these things. So total DNA isolation. So when you take a plant, when you take animal and when you take a bacterial cell, all these have a different type of cell wall material, and the plasma membrane composition also will be different. So depending on that you have to use different lysis buffer, and plant tissues also will be having some amount of flavonoids, phenolics, colored pigments. So because of that you have to you have to exchange buffer, or you have to do extractions several times to get a good quality DNA from all these different organisms. So because of that, cell wall composition is different, the cell cytoplasm materials are different. So because of that, all in these organisms, the protocol for isolating total DNA is different. So in the next video, we will see how you can isolate genomic DNA from plant, how you can isolate genomic DNA from animal cells, and how you can isolate DNA from bacterial cells and viruses. Thank you.